Welcome to The Explainer. Today, we're going to go way beyond what you probably learned in high school biology. We're diving into the wild, fascinating, and sometimes really strange rules of inheritance that prove genetics is so much more than a simple Punnett square.
Okay, so let's start with the basics we all know, thanks to Gregor Mendel and his pea plants. You've got a dominant gene version, the allele, and a recessive one. The dominant one pretty much calls all the shots, while the recessive one kind of gets pushed to the background. Seems pretty straightforward.
But then, nature loves to throw a wrench in the works. What about this? You take a red flower, cross it with a white flower, and you get a pink flower. So, wait a minute. Which one is dominant? This little puzzle totally breaks that classic rule we just talked about. So what on earth is going on here?
Well, that question leads us right to our first big stop beyond Mendel. We're going to look at the really cool ways alleles can interact when there isn't one clear winner. It's less of a fight for control and more like a creative collaboration.
And the answer to our flower puzzle? It's a concept called incomplete dominance. And it's pretty much what it sounds like. The dominant allele just doesn't have enough juice to completely cover up the recessive one, so you end up with something in between.
Let's put these side by side. You see, in that classic model, red would just win. The flower would be red, end of story. But with incomplete dominance, you get a blend. The easiest way to think about it is like mixing paint. You mix red and white, you get pink. The traits are literally blending together to make a whole new look.
Okay, but what if instead of blending like paint, both traits just decided to show up at the party, at the same time, in their full glory? Well, that's not blending. That's more like sharing the spotlight. And it has a name, codominance.
And the perfect example of this is actually running through our own veins, our blood types. Someone with type AB blood doesn't have some weird blended in-between antigen on their red blood cells. Nope, they are fully expressing the A antigen and fully expressing the B antigen at the same time. Both alleles get equal representation.
All right, so we've seen how genes can blend and share, but now we're going to move into some more extreme territory, where certain genetic combinations aren't just about looks, but can actually be a matter of life and death.
A whole bunch of our genes are just absolutely essential for survival. And if you happen to inherit two broken copies of one of these critical genes, well, it can be fatal. This is what we call a recessive lethal allele. The danger is hidden if you only have one copy, but it's expressed if you get two.
A classic example of this is the curly wing allele in fruit flies. If a fly gets one copy, it just has these kind of cute curly wings. No big deal. But if a fly inherits two copies, it's lethal. The fly never even develops. This totally messes with the expected ratios. When you cross two curly wing flies, you don't get a 3-to-1 ratio. You get a 2-to-1 ratio of curly to normal winged flies among the ones that actually survive. A whole quarter of the potential offspring are just gone.
Now there's an even rarer and honestly more frightening pattern, a dominant lethal allele. With this, just one single copy is enough to be fatal. So you have to wonder, how could an allele like this even get passed on? If it kills you, how do you have kids? Well, the whole secret is in the timing.
The only way these alleles stick around is if their deadly effects show up late in life. And Huntington's disease is a truly tragic example of this. The fatal breakdown of the nervous system usually doesn't start until a person is well into adulthood, often after they've already had children. And this creates this absolutely heartbreaking situation where someone can pass the allele to half of their kids without even knowing they carry it themselves.
Okay, so everything we've talked about so far has been about the genes found on chromosomes, all packed away nice and neat inside a cell's nucleus. But what if I told you that's not the only place you inherit DNA from?
Yeah, this is something called extranuclear inheritance. It has to do with the tiny little loops of DNA found in our mitochondria, the powerhouses of our cells. And since this DNA lives outside the nucleus, it plays by a completely different set of rules than Mendel's.
So why is this a big deal? Well, think about it. During fertilization, a sperm cell basically just drops off its nuclear DNA. That's its main contribution. The egg cell, on the other hand, provides all the cytoplasm and all the machinery for the new embryo. And since the mitochondria are floating around in that cytoplasm, you get all of your mitochondrial DNA from your mom. Every single bit. It means certain traits and diseases are passed down only through the maternal line.
To keep track of all these different, sometimes confusing patterns, geneticists use family trees called pedigree charts. The symbols are really simple. A circle is a female, a square is a male, and if the shape is filled in, it means that person has the trait we're looking at. They're an amazing tool for seeing how a trait moves through a family.
And these pedigrees are also super helpful for spotting other parent-specific patterns, like X-linked inheritance. Remember, males are XY and females are XX, so a son always gets his X chromosome from his mom. This creates some really obvious clues on the family tree, like the trait showing up way more often in boys, and a dad being physically unable to pass it on to his son.
All right, let's get to our final topic, and honestly, this one might just be the most mind-bending of all. It's the idea that some of our genes actually remember which parent they came from.
This is called genomic imprinting, and it's what we call an epigenetic thing, which means the actual DNA code doesn't change at all. Instead, the cell sticks a little chemical tag on the gene, basically silencing it. It's like putting a do not read sign on one parent's copy of a gene. So even though you have two copies, you're only using one of them.
Now, this silencing tag isn't permanent. It follows a very precise life cycle. The imprints get put on when sperm and eggs are made. They're kept on in all the cells of the new baby, but then they get totally wiped clean in that baby's own reproductive cells, so the whole process can be reset and start all over for the next generation.
And this right here is where you can see just how powerful imprinting is. Prader-Willi and Angelman syndromes. Two very different disorders, but they're both caused by a problem in the exact same stretch of DNA on chromosome 15. The only difference, Prader-Willi is caused when you lose the working copy from your father. Angelman syndrome is caused when you lose the working copy from your mother. It's unbelievable proof that who you inherit a gene from can be just as important as the gene itself.
So wow, from flowers that blend their colors, to blood types that share, to genes that can be lethal, to DNA we only get from our moms, and even genes that have a parental memory, it is so clear that the world of genetics is way richer and more complicated than those simple rules we started with.
The real takeaway here is that our inheritance isn't just a static blueprint. It's a dynamic, layered story. And that leaves us with a pretty deep thought. If our genes can carry a memory of which parent they came from, what other kinds of stories are we inheriting without even realizing it?